Can you see me okay? Yay. Hi, I am Mrs. Sloan, and this is Honors Biology, and this is our Unit 2. So I'm going to get us going here, start the class, and go here to our top 10 items that we need to know for Unit 2. So just take a minute to review those items. See if there are things maybe you feel strong about. You can star them. Feel a little weak, maybe you highlight them, whatever you want to do. Now I want to remind you for your final, am I going to put any additional objectives on the final? No, I am not. 0 additional objectives on the final, but this is a review I made for like when we were doing Unit 2. So that's why they're there. That's a big unit, huh? It's way big. So we'll focus on just maybe the hard stuff. So if you're feeling good about something, you're going to go like this so I can move on, right? That'll be the signal. And we'll move on from it. Okay, so plate tectonics, just knowing the parts of the earth. We good on that? Okay. And then we talked about what moves the plates on Earth, and those are what? Convection currents is what you want to know there. And then you just want to know what are the different conditions you can get, right? Convergent, they come together, divergent away. And what you get as a result of that depends if you've got 2 continental plates. If you have 2 continental plates coming together, what are you going to get? A mountain, right? It's like having 2 semi-trucks coming at each other. It's only going to go up, right? But what happens if you have an oceanic plate converging on a continental plate? Yep, subduction zone. But what's going to then happen, right? Because that oceanic plate goes under the continental plate, what do you tend to get? Volcanoes. Good. And that's why our oceanic plates are fairly young, relative to our continental plates, because our continental plates stay. It is our oceanic plates that can go through subduction and then get reformed again. So we talked about that so we would understand a little bit about the Earth's process, but it's also, we need to have the evidence of that. So the evidence of plate tectonics, we could measure the age of the Earth coming out at a divergent plate boundary, right? And we could look at that, like the mid-Atlantic ridge, and we can see that the farther you get away from the mid-Atlantic ridge, the older the rock is. The closer to the mid-Atlantic ridge, the newer. But also, when things like crystallize, when you have those iron, right, when it becomes solid, it's going to depend on north and south. So we can see the alternating north-south in a pattern. We also saw other pieces of evidence, like the uniformity of fossils when all the continents were together. What's that called? Pangaea. And this right here is biogeographical evidence for the unit we're on right now for evolution, right? This, okay? That's our biogeographical evidence, because 65 million years ago, the plates were separate from each other, and that's about the time that mammals were evolving. So that's why we have such unique mammals on each of the different continents, because that's when they started developing. We can also see the continuity of mountain ranges if you put them together. And sedimentary rock layers. You can see the continuity if you put like Africa and South America together. All right, so that was our evidence for it. And then the other thing we kind of addressed was just the idea, there it goes, it's not going in on mine, but there it is that how life on Earth could influence the land, and then the land could influence the life, right? If the land moves, like what you saw in Antarctica, then what can live on there is going to be altered. If you have volcanic activity, what can live there is going to be altered. And a really good example of that is the proliferation of oxygen and the oxygen revolution about 2.3 billion years ago. So life, plants evolved the ability to water. What are we going to do? Break it in half. When you break it in half, what do you get? Oxygen. So plants at first, autotrophs, didn't know how to do that. And later, when that adaptation evolved, they could split water. Then that oxygen changed the environment. It made it an oxidizing environment instead of a reducing environment. things would break down instead of build. And at first, oxygen was considered a poison and most life was killed. But those who still survived, they could use oxygen in cellular respiration. And what do we call that when you use oxygen in cellular respiration? Aerobic respiration, right? Then that was great because you maximize the amount of ATP that you have. Okay, so that's life affecting Earth and vice versa. All right, so then we talked about the origin of life, and early Earth again was missing what? It was missing oxygen. So it was a reducing environment conducive to building. Now, do this part with me. This will help you, okay? I don't think I did this in class. So how old is the Earth? About 4.5, 4.6 billion years old, right? And the plates are moving around. Move your plates. Make a subduction zone. What are you going to get? A volcano. So now gases are out there on the Earth. What kind of gas? What are they missing? Oxygen, because it's a what? Reducing environment. But there's a lot of energy here, right? Where's the energy on the early Earth coming from? What is that? Lightning, UV radiation, molten magma, and then isotopes, radiation from the Earth's core. So these gases right here are, what else are you going to have? You're going to have some rain, right? And you're going to have oceans. That early Earth, the gases in those oceans would have been just gases that came out of a what? Volcano. But you have all of the energy that we talked about, and those early gases then could have gone from inorganic molecules to what? Organic molecules. We saw an example of that. Who did an experiment where they showed that that could happen not in millions of years, but a week or so? Miller. Miller did an experiment where he did that. Yes. Organic molecules have carbon, more than one. So carbon monoxide and dioxide are not considered organic. They're too simple. Okay. But anything else with carbon in it is considered organic. Okay. Exactly right. So these are all inorganic molecules right here, right? Because CO2 and carbon monoxide are exceptions to that. So our four important organic molecules are what? What are our four important organic molecules? Carbohydrates, proteins, lipids, nucleic acids. What do we use? We've already learned this to catalyze reactions. Enzymes, right? Enzymes catalyze reactions. Do we have enzymes to catalyze these reactions to build these things? No. So where did the energy come from to facilitate these reactions? Lightning, UV radiation, molten magma, radiation. The same thing that helped us go from inorganic to organic helped you go from monomers to what? Polymers. Okay, monomers to polymers. And then that's all within this unit is, sugars build carbohydrates, amino acids build proteins, fatty acids build lipids, nucleotides build nucleic acids, right? Those are monomers building polymers. Then these polymers are what's going to build your cell. Now, what do all cells have going around the outside? Cell membrane that decides what goes in and what doesn't, right? What is a cell membrane made out of? Phospholipid bilayer. And embedded in it, because this is part of this unit, you have what? Proteins. Those proteins, do this with me. This is part of this unit. Bye, Nikki. Thank you. Channels, carriers, cell recognition, receptors, enzymatic junctions. Okay? So when you have these macromolecules, you have the beginnings of cell membranes because you have both lipids and proteins who can build that barrier. So what's going on inside that cell, protocell, is different than what's going on outside, right? The first cell, prokaryotic, eukaryotic. Which one? Prokaryotic, the simple one, right? First cell, autotroph or heterotroph? Heterotroph. It's easier to just consume nutrients than it is to build them through the process of photosynthesis, right? So first cell was probably a prokaryotic cell. It was a heterotroph. The oldest fossilized cell is how old? About 3.6 billion years old, right? So you had 1 billion years from the time the Earth formed to go up these steps to make a cell. 1 billion years to climb those steps to form the very first cell. All right. Then, yes. The key inorganic molecules, not all these right here, but I would know these. I taught them to you. What's this? Oxygen. Those are the 3 inorganic molecules I would know. Okay, anything else on that? All right. So then we transitioned from, okay, so we have cells. So then we kind of looked at the chemistry of life. And so we looked at the structure of an atom. Let's do the song. Ready? Here we go. The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8, 8 is great, octet rule. So let's break it down. The atom story. Atoms are made out of what? Protons. What do you call the proton number? Atomic number. Can you change the atomic number of an element? No. That is what defines it. Hydrogen, atomic number 1. Helium, atomic number 2. Every single time. If you change the number of protons, you are changing the element. Okay, protons, neutrons. Ooh, can you change the neutron number? What is that called? Isotope, right? An isotope. What else will that change about that atom if you change the number of neutrons? Atomic mass. Because the atomic mass is the unchanging number of protons plus the changing number of what? Neutrons, which will affect your atomic mass. You okay so far? Protons, neutrons, and electrons. Electrons hang out in those energy levels. Very first energy level can hold how many electrons? 2. Hydrogen has 1 electron in its outermost energy level. That's it. That's what makes hydrogen so reactive. Because the number of, what do you call it? Your outermost energy level is called your what electrons? Valence electrons. The number of valence electrons you have determines your reactivity. Because elements are trying to get happy. There's 2 ways to get an element happy. Fill up, right? What? Fill up what? Energy levels. Fill up its energy levels or get it to 8. So is hydrogen innately happy? No, it has 1 valence electron. Helium, though, has 2 valence electrons. Is he happy? Yes. He is referred to as a what kind of gas? Noble gas, because he doesn't need to interact with anybody. He doesn't need to share, ditch, anything. Question? So the octet rule is not going to apply until you're up in the second energy level, because the first energy level only holds 2. And there's only 2 elements who fall under that. All the elements are above that. Okay? All right. So then that's what we did, is we would look at different elements like lithium. And I know that the smaller number is the atomic number, right? The number of protons. The bigger number is the mass. There's nothing you have to memorize about that, but you just think mass, you would have protons plus what? Neutrons, right? Is lithium happy? No. Lithium has how many protons? 3. So it has also 3 what? No. It has 3 what? Electrons. How many can you fit in the first energy level? 2. How many do you have left? So it's going to be out there. That second energy level holds how many? 8. So is lithium happy? No. So what are lithium's options? Okay. It could ditch an electron. Do you agree with that? It's not too hard. If lithium ditches one of its electrons, it has 1 less negative charge. That means overall it will have a what? Positive. What do you call it if you have a charge like that? When you vary the number of electrons, it's called a what? Ion. Now lithium, because he gave it away now to get happy, to go down to the next energy level to get happy, he has a positive charge. Who is he going to be attracted to? Somebody who has a negative charge. And what kind of bond do you call that? An ionic bond. That is one way for lithium to get happy. Ditch that outermost electron. Okay? What would be another way for lithium to get happy? It could share. It could share with somebody who has 7, and then his one outermost electron that he has out there could be the 8. And 8 is great. Okay? So he shares with somebody who has 7. They're both happy. It's a win-win. Would you expect lithium to be able to steal 7 electrons to get to 8 and become a negative ion? No, there's no way it can steal 7 electrons. It doesn't have the power to do that. No element's not going to steal 7 electrons, right? If you only have a few, you're probably going to share or give it up. If you're pretty close to 8, you're probably going to steal or you're going to share. Those are your options, right? Okay, so when we look at the periodic table, remember, periods are on the horizontal because when you're on your period, you want to lay down, in case you're wondering. Okay. And they are at the same energy level. #tired. Okay. But on the vertical, you call them what? Groups. And so when you look at the periodic table, the same groups, right? If you're in the same group, you have the same number of valence electrons. So therefore, you will behave the same way. Everybody in the group behaves the same way. Everybody here in group 1 has 1 valence electron, so they behave like that. They either give it up or they share with somebody who has like 7. Does that make sense? Okay, but if you're in group 2, you might give it up or you might share with somebody who has what? 6, because that'll bring you to 8. Okay, so those are the patterns that you can see in the periodic table. We already talked about isotopes, when you gain or lose an electron. Gain or lose a what? Neutron. Okay, neutron. Isotopes vary the neutrons. Okay, we talked about already ionic and covalent bonds. When you share in what? Pairs. So if you have a single bond, you're sharing 1 pair of electrons, you share 1, they share 1, right? Do you always share fair? No. What do you call it if somebody has a stronger pull on those electrons? Polar. If you share exactly perfectly fair, you are what? Nonpolar. Okay. Where does this come into play? Water. And that's because living things are mostly made out of what? Water. So water's in our cells. Our cells are sitting in water. Water's super important. And because oxygen is very electronegative, has a strong pull, and it has 6 valence electrons, it really pulls when it shares with hydrogen, who's like, okay, it's like, whoa, okay. And oxygen becomes partially negative in a water molecule, and hydrogen becomes partially positive. Hydrogen bonds are between 2 different water molecules or anything that has that kind of charge, okay? 2 different, okay? Different water molecules between different molecules. That's what hydrogen bonds are. You don't, but here, okay, holding this oxygen and this hydrogen together, this bond right here. No, I can't. Maybe. That right there that I'm trying to highlight with the arrow, that is a polar covalent bond, right? Polar covalent bond. Okay, perfect. All right, take a breath in, let it out. You can survive. So then we have all of those properties of water. Like it has a high heat capacity, a high boiling point, because it can hold all that energy in its hydrogen bonds, right? We looked at that. It's a good solvent if you are hydrophilic, not if you are hydrophobic, right? It's a liquid instead of a gas. I mean, think about it. Hydrogen is a gas, oxygen is a gas, but together they are a liquid. That's because of all the hydrogen bonds between the water molecules. I already did that. Less dense when frozen. This is really good for life in the water because that ice forms that nice insulating layer. Now, pH, because solutions dissolved into water, water is so important to life, you get a whole bunch of hydrogen ions dissolved in your water, you are called a what? If you have a whole bunch of hydrogen ions dissolved in your water, you are a what? An acid or a base? you are an acid. Okay? So it's always in ratio acid, hydrogen ion to hydroxide, OH minus. What is H plus OH? H2O, water, neutral, right? If it's the same amount of H and OH, you're neutral. More hydrogen ions, you're acidic. More hydroxide ions, you're basic. The thing about pH to remember is one number is a big deal, right? Outside right now, I could see what the weather is. Let's say it's 67. If we find out I'm wrong and it's really 68 outside right now, is that any big deal to you and I? No. But in pH, it's a big deal because every change is how many? 10. Good. All right. And then buffers help to keep things at the right pH. Okay. Then we switched to organic molecules and carbon. We talked about how carbon can build 4 bonds, right? Because it has six electrons, 2 on the first energy level and 4 on the second. So it wants to make 4 bonds. We talked about the building bonds are called dehydration, what? Synthesis. So you remove an H and an OH to form water, right? But you hook them together. And then what do you call the breaking apart one? Hydrolysis. And so that's the building. This is the breaking. We talked about the importance of enzymes, that enzymes are specific to their substrate. We actually learned that later, but they catalyze all those reactions. We then went into a whole thing about the important organic molecules, right? Sugars build our carbohydrates, and polysaccharides, we have structural and we have energy. Energy is things like starch and glycogen, if you recall. Okay. And structural things like cellulose and chitin. We talked about lipids. Lipids are either fats or steroids. Fats are glycerol, 3 long fatty acid chains. Those fatty acid chains could either be what or what? Saturated or unsaturated. And saturated, the chains are straight because it's saturated with hydrogen. There are no double bonds. But unsaturated chains, they have kinks in them because you have double bonds. Animal fats are what? Saturated fats, and at room temperature, they are a solid. Whereas plant fats are a liquid and they are unsaturated fats. A modification of a fat is a phospholipid we find in cell membranes, right? A phosphate group, just like adenosine triphosphate, ATP, that phosphate group we've been talking about, attached onto glycerol and 2 fatty acid chains. You can also have another kind of another kind of lipid. You can have steroids, which are 4 rings. We find steroids in cell membranes. They're called what? In animal cells. Cholesterol. Good. Okay. And then we talked about amino acids build proteins. Remember that? And we talked about how many different R groups are there? 20. Good. Right? We talked about nucleotides, which are a base, a sugar, and a phosphate. And those will build nucleic acids like DNA, ATP, and RNA. Okay. And then here, ATP, I already got that. Then here is that chart. Remember this whole chart? But yet there was more. We started talking about cells then. Remember the cell theory? All living things are composed of cells. Cells only come from pre-existing cells. Cells are the functional unit of every living thing, okay? We talked about that cells are very, very, very what? Small, because they want a large surface area per volume. We compared and contrasted prokaryotic cells to eukaryotic cells. The big thing about prokaryotic cells, they have no membrane enclosed organelles. They have DNA, but it's not enclosed within a nucleus, right? It's just a nucleoid region, an area. They have ribosomes. They can do cellular respiration, they just don't do it inside of a mitochondria. They can do photosynthesis, they just don't do it inside of a chloroplast. They don't have any compartmentalization. Yes, we don't have to know that right now. It's an interesting topic, but we still sorry. Okay, so then here I kind of went over like the cell membrane decides what goes in and out. You already know ribosomes are the site of what? Protein synthesis. Flagella is for movement. Fimbriae help you attach. Did I teach you sex pillars, right? Yeah. Okay. Then we said, okay, there are 3 domains. We've talked about this before. Archaea and bacteria are both prokaryotic cells, but archaea are called what? Extremophiles. Extremophiles. They can live like really salty or high temperature or low pH. Then eukarya, and we reviewed this last time, right? The different kingdoms, but they are all composed of eukaryotic cells. And then the last part of this was just knowing the different organelles and what they do, which you can just review using a chart, right? Okay. And looking at differences between like an animal cell and a plant cell. Like a plant cell has a cell wall, doesn't have any centrioles, has a large central vacuole, it has a chloroplast, right? And you just review what each of those do. Okay, and then that brings us to our last chapter in this unit. Okay, and that was all about the cell membranes. Let's do some songs to remember that. I think some of you helped me with that before, right? The cell membrane, right? We already talked about the proteins that are in it, right? So who can go through a phospholipid bilayer as long as you are not what? Large or charged. If you are large or charged, you're going to have to use a what? Channel or a carrier or whole membrane, which would be endocytosis or exocytosis. 3 types of endocytosis. Pinocytosis, phagocytosis, cell eating, or ding, 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 ding. Yep, receptor mediated endocytosis. Do you remember those? Pinocytosis, cell drinking, phagocytosis, cell eating, receptor mediated, they like bind to receptors, and then it does endocytosis. There is only one kind of exocytosis. Yes? Yes, it would be a subtopic of pinocytosis. Exactly. Okay. Now, we also talked about when you go through a phospholipid bilayer, let's say you're going with the concentration gradient, right? Whether you go through the phospholipid or you use a channel or a carrier, okay? When you go from high to low, is it going to cost you any energy? No. What is that called? What kind of transport? Passive transport. Okay. Here, let me give you a slide. This is just the structure of the cell membrane. Just remember phospholipids, proteins, cholesterol. Remember glycolipids and glycoproteins, sugars attached to the outside, either attached to a protein or lipid. And then the cytoskeleton is here. Okay, so if you're just going from high to low, okay, then that is passive transport. It's not costing you any energy. If you go through the phospholipid bilayer, straight up diffusion. But if you have to use a channel or a carrier to go to high to low, then it's called facilitated diffusion. It had to get facilitated by a channel or a carrier. It couldn't go through, probably because it had a what on it? Charge. Yeah. Active transport is when you go against the gradient. You have to use energy to concentrate something to one side of the membrane, like hydrogen ions to one side of the membrane so you can generate ATP. Yes? Okay. Then we went into the whole topic of osmosis. Remember osmosis? It has 3 parts to it. Osmosis is the diffusion of water across a semi-permeable membrane. And water must flow from the hypo. So it's going to flow from where it is more concentrated to where it is less. If you have a hypertonic solution, that means you can think of like somebody who's hyper, they have a lot of sugar, okay? And one over here, it's not pure water. It's got some things dissolved in it, but it's got more water on this side than that. And it's always relative to each other. If this is up, this is down. If this is left, this is. If this is hypotonic, the other side of the membrane has to be what? Hyper. And water will flow. What if it's same? What is that called? Isotonic. Okay, and we looked at some different scenarios with red blood cells, right? This red blood cell is in an isotonic solution, this plant cell. So the same amount of water flowing in is also flowing out, okay? This red blood cell is sitting in a hypotonic solution. Around it is hypo. Water must flow from the hypo. It's going to go into that red blood cell, and it can make it what? Burst. Will it do that to a plant cell? No, because it has a cell wall. Yeah. In this case, it's in a hypertonic solution. So if this is hypertonic, the other side of the membrane is hypo. So it will flow out and it shrivels. And it shriveled there. So we talked about like carrots and things like that. And if you wanted really crunchy carrots, what should you soak your carrots in if you want really crunchy carrots? Pure distilled water. Because that pure distilled water will be hypotonic. The carrot would be hyper. Water will flow into the vacuoles and push against the cell wall, and you will have crunchy carrots. Okay, and then this is just the forms of endo and exocytosis right here. Endo, there's your receptor mediated. And there's that. And oh my gosh, the end. You did it. Good job. Good job. Hang on, let me just stop this real quick.